Hi guys, and welcome back to our cellular biology series, part 2. In this exciting episode, we're taking our exploration of the microscopic world to the next level. Get ready to delve into the realm of cellular dyes, where we will uncover the secrets of how these colorful agents helps us decipher cellular mysteries. But that's not all. We will also introduce you to the incredible array of optical microscopes, the tools that bring those vibrant cellular landscapes into sharp focus. So, whether you are a biology enthusiast or simply curious about the hidden universe within, join us on this journey of discovery. Let's dive in. Dyes. What are dyes? A dye is a reagent capable of coloring a substance or a group of substances in a lasting manner. It must present two properties: possess groups which give it a color, and they are called chromophores, and have groups which ensure its permanent fixation on the structure to be dyed, and they are called oxochromes. So let's talk now about oxochromes. We have two types of oxochromes. We have acid dyes and basic dyes. For acid dyes, they are also called cytoplasmic dyes. An acid dye is negatively charged and exhibits an affinity for positively charged substances. And then basic dyes. A basic dye is positively charged and exhibits an affinity for negatively charged substances, acids. Now let's move on to coloring categories. We have two coloring categories. We have topographic coloring and special coloring. For special coloring, we use special colorants to color four types of substances. We have connective fibers, carbohydrate substances, lipid substances, and proteins. For connective fibers, we use the Van Gaten method, and we have the Masson's trichrome method and Gomori's method. For carbohydrate substances, we use PAS, Congo red, and Alcyon blue. For lipid substances, we have red, oil method, and black Sudan. For the proteins, we have the Graham-Vigert method. For cytological samples, for cytological samples, we are going to study two examples. The first example is going to be about blood smear, and the second one is going to be about oral smear. So let's start with the blood smear sample, cytological sample. The principle: the blood smear consists of carrying out a single cell spread of blood elements. The material that we need to make a cytological sample: blood smear, between brackets, 2 blades, a sterilized needle. Operating mode: using a sterilized needle, prick the tip of a finger, place a drop of blood 3 millimeters on the end of a slide, with the second blade held at 45 degrees to the first. Touch the drop of blood, then spread it with a brief movement on the first blade to obtain a fine spread. Dry immediately by shaking the blade. And here is an image that shows the operating mode. So now let's move on to the second example, oral smear. An oral smear is categorized under the exfoliative cytology. And what is exfoliative cytology? Exfoliative cytology studies the cells that naturally detach from an epithelium. Vaginal smears, oral swabs, cells of the bronchial mucosa in sputum. So the advantages of oral smear or of exfoliative cytology: non-aggressive method, easy to perform in the mouth, gives rapid results, used for the diagnosis of different oral conditions, in particular to identify certain specific conditions on the oral mucosa. Disadvantages: often does not allow a definitive diagnosis to be made. The material that we need: a clean and sterile histological slide, a fixing solution, alcohol, ether, ethylene, a tongue depressor. Operating mode: place a damp compress on the injured site, elimination of mucus. Scrape the surface of the lesion several times using a tongue depressor or a metal spatula or other. Spread the scraping product on the blade. Fix immediately, then coloring with H&E or methylene blue. Speaking of the fascinating world of staining and cellular visualization, let's now shift our focus from blood and oil smears to the incredible technology that enables us to explore these microscopic wonders in even greater detail. We have seen how stains can provide crucial insights into cellular structures. And now let's dive into the optical microscopes that serve as our windows into the captivating realm. The different optical microscopes. We have bright field microscope, phase contrast microscope, fluorescence microscope, confocal microscope, dark field microscope, polarized light microscope. Is very little used in biology. We have reflection microscope. So let's start with bright field microscope. A bright field microscope is a normal microscope, upright microscope, used in transmission. Applications: histology, hematology, parasitology, and bacteriology. Phase contrast microscope facilitates the examination of non-fixed, non-stained specimens with topographical features such as cell spreading and cell cultures. Fluorescence microscope enables the visualization of both endogenous and notably exogenous fluorescent molecules. The light source is positioned laterally and comprises a lamp with an extended wavelength spectrum. Reaching into the UV range, an excitation filter is employed to choose a specific wavelength range tailored to the fluorochrome of interest for visualization. So now, confocal microscope. The advantages of confocal microscopy over traditional microscopy include enhanced image resolution and the capability to perform optical sectioning, allowing for a sequential examination of the specimen plane by plane. Subsequently, these individual planes can be amalgamated to reconstruct a three-dimensional image. So, now we have the dark field microscope. Illumination is achieved through a dark field condenser, ensuring that no light directly enters the objective lens. This configuration finds application primarily in the field of bacteriology. Then we have polarized light microscope. Allows you to observe thin slices of rock and teeth and bones not decalcified. Then we have the reflection microscope. This method is employed when dealing with opaque samples, allowing for the observation of light that is reflected from the sample's surface. It is particularly useful in examining the microstructure of materials following sample polishing and chemical treatment. So today we delved into the fascinating world of cell coloring categories and explored the various optical microscopes that help us unlock the mysteries of cellular life. We learned how cellular dyes and stains are used to highlight specific structures within cells, aiding in our understanding of their functions. We also journeyed through the diverse landscape of optical microscopes, from the traditional bright field microscope to the advanced confocal and dark field microscopes. Each of these tools provides a unique perspective on cells, allowing scientists to uncover intricate details and delve deeper into the microscope of life. Remember, whether you are a seasoned scientist or just curious about the microscopic world, these techniques and instruments are essential in revealing the complexities of cells and their inner workings. Thanks for joining us today, and don't forget to like, subscribe, and hit that notification bell for more exciting explanation into the world of science and microscopy. Until next time, keep observing, keep learning, and keep exploring. Bye.